Gregor Mendel came up with two fundamental principles of heredity. He came up with the law of segregation, and he came up with that when he looked at one gene at a time. And then when he looked at two genes at a time, he came up with the law of independent assortment. Well, that law of independent assortment, when we looked at it in chapter 14, we were making the assumption that the genes were going to be located on different chromosomes. So if we review meiosis and why the law of independent assortment works, if you look at this picture here, we've got two genes that are located on different chromosomes. We've got a green gene, or an A gene, that's located on the small chromosome, and we've got a blue gene, or the B gene, which is located on the large chromosome. Now, considering that each one of the tetrads that forms during meiosis can line up independently on that metaphase plate, that does result in different combinations when we look at the gametes later on. So we could get both of the dark colors lined up on one side, and that would cause us to get capital A, capital B, and lowercase a and b. Or we could get a light color and a dark color lined up together, and we get something different at the end based on that. Now, each round of meiosis is going to be independent assortment, or independent lining up. So when all is said and done, we expect that 25% of our gametes are going to be like this, 25% like this, and so on the whole way across. And that's why when we draw a Punnett square with gametes from this particular parent, we would just list all four of those possibilities across one side of the Punnett square. Now, if we review Mendels' experiments, he did look at a number of seed characteristics. He did look at seed color and seed shape, which is the example we're looking at here. And from this parent, we would expect that we would get any combination of the Y's with the R's. And again, that's based on his law of independent assortment. So we would expect to get the capital Y with the capital R, the capital Y with the lowercase r, little y with big R, and little y with little r. And those are the four possibilities. We expect to get 25% each one, which is why over here we list those across the top of the Punnett square. From this parent, there's only one possibility, and that's going to be lowercase y, lowercase r, and that's what we put on this side of the Punnett square. So if we go through and we fill in the boxes, we look directly across, directly above, just as we did before. We can fill in the boxes, and in addition to that, we can also assign the phenotypes of the individuals. So notice when we look at the individuals after we've assigned the phenotypes that you do have the first two boxes here, and these look like the parents. These are what we call parental offspring. We also have some offspring that have new combinations of these parent traits, such as yellow wrinkled and green round. We call these the recombinant offspring. So we're taking these offspring and we're dividing them up into two groups based on the combination of traits that they have. And the recombinant ones will have the new combinations that we haven't seen before. In Mendels' experiments, he got results that matched up with the ratios he would predict from the Punnett squares. And notice that that means that he had equal numbers or equal amounts of parental offspring versus recombinant offspring. So essentially, 50% of his offspring were parental and 50% of his offspring were recombinant when he did this cross. Now, as he was working with the pea plants, we said that Thomas Hunt Morgan was repeating a lot of his experiments in fruit flies. And Thomas Hunt Morgan, after he finished looking at one gene at a time, he also went on to look at multiple genes at a time and to study the inheritance of that. So he did have a number of different characteristics that he had identified in fruit flies where he had some variation there that he could compare. He had a body gene, and this one also had a mutant version. So we have B plus and we have B. B plus was for gray, and then he also had the B, which was for black. He had another gene, which was a wing type gene, and they had short, tiny wings in the mutants, so he called them vestigial wings, and I'll just say short wings here. And then he had the normal wing type. So he took a fruit fly that was homozygous wild type for both of the genes. So this one had a gray body and it had normal wings, and he crossed that fruit fly with the double mutant over here, which was a black body with the tiny wings or vestigial wings. When he did that cross, we know from this parent we're going to get B plus and VG plus, and we know from this parent we're going to get B with the VG. So we are going to get this F1 down here, which is heterozygous for both of the genes. He then performed a test cross, very similar to what we just looked at on the last slide with Gregor Mendel, by crossing that F1 hybrid with the double mutant again. So from this one, we would expect to get any combination of the B's with the VG's, so B plus with VG plus, which is the first one here. We expect B plus with VG regular, which is over here. We expect to get B with the VG plus, which is this one, and then we expect to get the two mutant versions together, which we have right here. From this one, we know that we're always going to get the B with the VG regular, so that's listed down this side. So we can go through, we can fill in the boxes just as we did before, and once those boxes are filled in, we can assign the genotypes. As long as they have a capital, or as long as they have a B plus, they're going to have a gray body, and if they have VG plus, they're going to have the normal wings. Now, if we look at these closer, we can also identify which ones are going to be the recombinant offspring and which ones are the parental offspring. Well, the first two boxes here will be our parentals, and these over here would be our recombinant ones. Okay, so we've got two boxes representing parentals, two boxes representing recombinants. And we would expect that basically we'd have 25% of our offspring in each one of these boxes. Well, if we look at the actual numbers that Thomas Hunt Morgan got, he definitely did not get 25% each one. So notice here, when we look at the numbers, he got 900 something of the first two boxes and somewhere around 200 of the other two boxes. So keep in mind, again, that these first two boxes are parentals and these second two boxes are recombinant ones. So he was definitely not getting 50% parental and 50% recombinant as Mendel was. So something else was going on here so that he was not getting independent assortment. Now, the reason he was not getting independent assortment is because he happened to be dealing with what are called linked genes. Genes that are located on the same chromosome tend to be inherited together in genetic crosses a lot of the times. So we call these linked genes because they are physically linked or physically connected to each other, meaning that they are actually located on the exact same piece of DNA, and because they're located on the same piece of DNA, they tend to go to the same place when we're talking about meiosis. This is going to alter the inheritance patterns that we expect, meaning we're not going to get the exact results that we would predict from a Punnett square. If you look at the meiosis shown on here, we're looking at one set of homologous chromosomes. We've got a B gene located on them, and pretty simple here, we would expect those homologous chromosomes to separate during meiosis 1, and then we would separate these sister chromatids during meiosis 2, so that we would expect that half of the gametes are going to get the capital B, half of them are going to get the lowercase b. Well, if we throw in a second gene here that happens to be located on the same chromosome, if, for instance, we put a capital A on the same one as the capital B and a lowercase a on the one with the lowercase b, since they're located on the same chromosome as the B genes, we would expect that they go to the same place. So in the end, we would expect the ones with the capital B to also have the capital A, and the ones with the lowercase b to have the lowercase a. So notice here, we're not getting the independent assortment or the any combination of the B's is equally possible or equally probable with the two combinations of the A's. So that's what we're talking about with linked genes. They're located on the same piece of DNA, and that will affect the assortment that we get in the gametes. Genes that are located close together on the same chromosome do not assort independently, which is what Thomas Hunt Morgan was beginning to find out. So we're not getting the expected ratios. If we're dealing with unlinked genes, these are genes that would be far apart on one chromosome or actually on different chromosomes altogether, they do assort independently. That is what Mendel was dealing with. So since Mendel was dealing with those, that's why his law of independent assortment worked out just fine. Thomas Hunt Morgan, though, was not dealing with unlinked genes. If you look over here, if we now assume that this B gene and VG gene are located on the same chromosomes, we would expect that this would be our F1 here. That one is going to have the B plus and the VG plus on one homologous chromosome, and then the two mutant versions will be on the other chromosome. So rather than really getting any combination of the Bs with the VGs, we expect the red chromosome to be passed on, or the lighter chromosome to be passed on. And notice that that really is the first two gametes that we have listed here. Those are the two gametes that will contribute to the parental type offspring. From the other parent, it's going to pass on the blue chromosome, so exact same thing there. Now, those are the offspring that we got a whole bunch of. They resulted from those single chromosomes being passed on together. However, notice that we do have some offspring over here. So what this means is that sometimes we do get these other two gametes produced. That means that there has to be a process that is going to enable us to get, for instance, this B passed on with this VG down here, or to get these other two passed on together in a combination, because we are getting some of those offspring. So if we do get some of the recombinant offspring, as we did, then this is incomplete linkage. So with incomplete linkage, there must be some process that occasionally breaks the physical connection between the genes on the same chromosome. So this really goes back to crossing over during meiosis. So occasionally we are getting a physical breaking of the DNA between these two genes. So if we go back and look here, when the tetrad is formed during prophase 1, occasionally we will get a breaking and a swapping of ends of the chromosomes between the B and the VG, and that's going to lead to these recombinant chromosomes. So we did call them recombinant chromosomes back there when we talked about meiosis, and recombinant chromosomes lead to the production of recombinant offspring, or offspring that have new combinations of the parent traits. So although we're not getting independent assortment, we are getting basically extra of the parentals and a few of the recombinant offspring. Now we can calculate what's called a recombination frequency based on how many recombinant offspring we have and how many parental offspring we have. To calculate the recombination frequency, we're going to take the number of recombinants, and so our percent is going to be the number of recombinants, so we add up our 206 plus our 185. And we're going to put that over the total number of offspring, which is adding together all of them, parentals plus the recombinants. So if we just write that out here to make sure we know where the numbers are coming from, then we're simply going to convert that to a percent, and we do come up with 17% in this case. So what that 17% means to us is that 17% of the time we do get a crossover event occur between the B and the VG genes. When we're looking at recombination frequencies, anything that is less than 50% means that we are dealing with linked genes. Our 17% is less than 50%, so these are linked genes. When we talk about linked genes, that means that these are most definitely on the same chromosome. And in addition to them being on the same chromosome, we can say that they are fairly close together. Okay, so they're close enough that most of the time they do stick together, because only 17% of the time are we getting a crossover between them. Now, if we're dealing with a 0% recombination frequency, that would be something that we would call completely linked. By completely linked, we never ever get a crossover between them. That means they are on the same chromosome and they are very close together. Now, if we think back to Mendel's example, in Mendel's example, we said that he had 50% recombinants, 50% parentals. Well, if we go and we throw some numbers in here so that we can calculate a recombination frequency, say we have 10 offspring of each one, just for simple numbers. For recombination frequency, you add up the number of recombinants, which would be these and these. So we've got 10 plus 10. And you put that number of recombinants over the total number of offspring, which is going to be 40 in this case. So if you look here, we've got 20 over 40, which is going to be 1/2 or 50% recombination. Mendel's genes were located on different chromosomes altogether. When you have genes located on different chromosomes, that is as far apart as they can possibly get from each other. That will give us a recombination frequency of 50%. So this means that our recombination frequencies should never go over 50%. That is the maximum amount that we get. So if you come up with a number like 70% or 80%, that should alert you to an obvious wrong answer in this case. So our recombination frequencies are going to range from 0 up to 50. And then based on what that number is, we want to have an idea about the location of the two genes that we're dealing with. If we look at an example here, we've got seed shape and color controlled by two genes. We're calling them G and H. And here we've got our cross. So I'm saying you've done this cross. Here are the offspring that you got, and we want to be able to interpret these results. So first off, if we're going to interpret these results, we need to be able to figure out which offspring are the parental types. By parental types, we're looking for ones that look like the parents. So if we just look at the ones that look like the parents, okay, we can just say, okay, well, the blue square, the red triangle. Even more simple than that would be to just roughly glance at the numbers that we have here. First off, the numbers are nowhere near equal, and we wouldn't expect them to be exactly equal, but we've got some that are in the 300s, the other ones are in the hundreds. Because we have numbers that are not equal to each other, the parentals are always going to be the high number ones. So you really don't even have to look at the appearance of them. We can just go straight off of those high numbers. So those are our parental ones. So if we put a little P here for parental, then the next thing we want to identify is which ones are the recombinant ones. Well, the other ones would be recombinant by default. Another way would be that they are the low numbers. And once we know that, then we should be able to calculate the recombination frequency. Okay, so if we do calculate the recombination frequency, you've got your equation up here at the top. We're going to take the total number of recombinants, and that should be easy since we've just identified which ones are the recombinants. We're going to take 166 plus 134, and then we're going to take all of that and we're going to put it over the total number of offspring, which will be our 342 plus our 166 plus our 134 and our 358. Then it's just a matter of taking that and converting it into a percent, which in this case, it ought to give you 300 over 1,000, which is going to be 30%. Now, that 30% number needs to mean something to us. So that 30%, first off, we want to know, does that mean that our genes are linked? And if they're linked, are they completely linked or are they incompletely linked? So remember that 30% means that we are dealing with linked genes. Not only are they linked, these are going to be incompletely linked. So incompletely linked would be anything that is greater than 0 and less than 50. Okay, and 30% certainly is. Because they are linked genes, we know they are on the same chromosome, and we also know that they are going to be fairly close together. Okay, so make sure that not only can we calculate the recombination frequencies, but we can interpret the meaning of the recombination frequencies. So if it is less than 50%, they are linked. We know that they are on the same chromosome, and we also know that they're fairly close together because most of the time they tend to stick together so that we have extra of the parental type offspring. Now, another reason that these recombination frequencies are so useful to us is because they can be used to come up with genetic maps. Genetic maps are ordered lists of the genetic loci on a particular chromosome. So in other words, they give us a picture of the chromosome, they show us which gene is located to all the other genes. Now you could get this same kind of information from genome sequencing, but genome sequencing is extremely expensive and time consuming, and there really is no way to sequence the genome of every single organism that is out there. So this is a very cost-effective way to sequence some of those organisms that people are not willing to spend quite as much money on as, say, the Human Genome Project. So what we're going to be coming up with are called linkage maps, and they're called that because they're based on recombination frequencies. They're going to provide us with a picture of the chromosome and give us an idea of where the genes are located at. Now, there is a correlation between distance apart that two genes are on the chromosome and the recombination frequency number that you get. Basically, the closer together that the genes are, the less likely they are going to have a crossover event occur between them. So in other words, the more likely they are going to be to stick together. So the closer they are, the lower the recombination frequency, and the further apart they are, the higher the recombination frequency. Now that is up to 50%, because keep in mind that we're not going to get a recombination frequency of greater than 50%, because 50% is what independent assortment gives us. Okay, so the further apart genes are on a chromosome, the more likely they are to be separated during crossing over, and the higher the frequency of recombination. Now, just to get an example, here we've got down here at the bottom what would be considered a linkage map. So we've got 5 different genes located on this chromosome, and we're seeing where those genes are located. We can see which ones are a little closer together than the others. And what we've done with our recombination frequencies is we've taken the percentage out and we've expressed the distances now in what we call map units. With these, 1 map unit is equal to 1% recombination frequency. So this is basically a relative unit of measurement. It's not a measurement that we could go and actually measure with a tool such as a ruler or something, but it does give us an idea of how close together or how far apart these genes are. So if we look on here, if we were to look at the recombination frequency between, say, this gene and this gene here, we've got basically 9% recombination frequency. So we have taken that and we have now changed it into 9 map units. Every time we calculate recombination frequencies, we are doing that looking at 2 genes at a time. So we look at those 2, we get 9%, it tells us 9 map units. If we were to look at, say, these 2, we would get the 48.5%, and we would convert that to 48.5 map units. Now notice that we've got from here to, say, all the way over here, and we're getting 67 map units. Now we can't simply take that and turn that into 67%, because keep in mind that we don't have percents over 50. So 67% would be most definitely wrong here. When you get a recombination frequency of 50%, 50% means 50 or more map units apart from each other. So that would actually, in that case, be 50% recombination. And the reason we would have this 67 over here is because we've got to add a bunch of data together to come up with that 67 number. So if we found out that from here to here was 48.5, we could figure out how far from here to here. That would tell us exactly how much distance between those, and then we can add it together and we can get the 67. The best way to really learn how to come up with these linkage maps is to go through a number of examples. I've got a number of examples to go through on here, and then you've got some other ones on your practice questions on Angel. So if we look at the first one, we've got 3 genes, A, B, and C, and we want to place these genes in the correct order on the chromosome. So I have provided you with the recombination frequencies. So we're assuming that you've done the necessary crosses to come up with this information. And there's different ways that you can approach these types of problems, but I'll just show you the way that I approach it. If you come up with another technique, that's fine. I always start off drawing a chromosome. So if we draw our chromosome here, and I always start with the genes that are located the closest together. So by closest together, I'm looking for the two genes that have the lowest recombination frequency. So A and C in this case, because we've got the 10%, these are most definitely the closest. So I take those two and I place them on my chromosome, A and C, and I know that they're 10% recombination, and that means 10 map units. So I'm going to mark me a distance of 10 right there. Then I'm going to go to the two that are the next closest, which will be C and B, and C and B are 35% or 35 map units apart. So if I locate my C on the chromosome, I know that B is going to be either 35 this way, or it could be 35 this direction as well. And since I don't know which way, that's the reason I'm writing it down below at this point. Then I'm going to look at my last piece of information, which tells me that A and B are 45 apart from each other. Well, if A and B are 45 apart from each other, that tells me that B must be located over here, because that'll give me the 35 I'm looking for there, and that will give me the 45 that I'm looking for up here. So when all is said and done, the order of these genes is going to be A, C, B, or you could also say that it's going to be B, C, A. Exact same thing there as far as our answer choices are concerned. So that's looking at 3 genes at a time. Now, if we throw in a 4th gene at a time, notice that now we've got more combinations, so we've got more recombination frequencies to deal with. Approach is really the exact same, so I'm going to draw me a chromosome here. And I'm going to start with the 2 genes that are the closest together, which is this right here, B and C, and they are 4. Okay, so I'm going to put my B and C right next to each other. They are 4 apart from each other. Then I'm going to go to the 2 that are the next closest, which will be B and D. B and D are 10%. So I'm going to say D could be here, or it could be 10 this way. Okay, when I go and look at C and D and the fact that it is 14 tells me that D must be located over on this side. So I can cross that out there. Then if I look at A and C are going to be 32. So that means that A is either 32 this way, or A is going to be 32 in this direction. If I look at A and B, and the fact that they are supposed to be 36, that tells me that this is the correct location for A, because that will give me 36 here, which I want to find. And then if I just double check it with the last piece of information, A and D ought to be 46, and that is going to work out to be 46 there. Okay, so the answer for this one would be D, B, C, A, which is also the same as saying A, C, B, D. Now, the order that you come up with out of those 2 options is just going to depend on how you wrote those first 2 genes on the chromosome. Keep in mind that inside the cell, there is no right and left, so either answer choice is just as correct as the other. If we look at one more example, it becomes a little more complicated if we throw in some 50% here. Keep in mind that 50% does not tell us an actual distance that the genes are located apart from each other. It tells us that they are 50 or more map units apart from each other. So if we approach this problem the same way we have done the other ones, we're going to start with a chromosome and start with the 2 that are the closest together, which is going to be A and D. So I'm going to put A and D right here in the middle, and I've got them 13 apart from each other. Then I'm going to go to C and D down here on the bottom. C and D are going to be 24, so that means that C is either this direction, 24, or C is this direction, 24. If I come up here and if I look at A and C is 37. So if A and C is 37, that tells me that this must be my C over here. So that'll give me 24 here, and it'll give me 37 right here. Then if I look at A and B are supposed to be 41. So that means that if we start where A is, B is either 41 this way, or it could be 41 this way. And now if I get to my 50%, if I look at B and C, for example, B and C, I got 50% here, and that means that it needs to be 50 or more apart from each other. So this B and C here, these two would be way too close. Okay, so it's not going to be that location for B. Instead, B is going to be way over here. Okay, because if I have B way over here, that gives me it 41 away from A. Sorry, did that wrong. Because that's not the correct C. Ignore that. Other direction. So if I know that my B and C are supposed to be 50%, then we've got